So diffuse intrinsic pontine glioma is the worst type of brain tumor. So basically every child who is diagnosed with it dies within 2 years. And so for me as a doctor who also takes care of patients with pediatric brain tumors, this is a very personal tumor for me because I've taken care of patients who have died from this. I've been there when they've died. I've been there at their time of diagnosis. Our project is looking at normal human neural stem cells and trying to use them to create models of diffuse intrinsic pontine glioma. In the last 2 years, there's been some really great genetic data that's come out that has identified a new mutation that is found in 70% of diffuse intrinsic pontine glioma. And our project, what we're trying to do is to take that genetic information and put it into the cells that we think go wrong and lead to this type of tumor. So we have normal human neural stem cells that are derived from the part of the brain that this tumor comes from, the pons. And what this project will do, will take that mutation that we think drives this tumor and put that mutation into these normal cells, try to turn these cells into diffuse intrinsic pontine glioma, and really investigate the basic biology of what does this mutation, which is found in 70% of this type of tumor, what does that do to normal cells? How does that begin the process of turning them into cancer cells? So we think that this particular mutation, which interferes with the way that the cells process their normal genetic signals, will freeze the cells in an undifferentiated state. So during the process of development, of going from a single fertilized egg to an adult, there's a lot of processes that have to happen where cells have to get signals to mature. And so what this mutation does is we think it blocks cells in an immature state and allows them to continue to grow and gain more mutations that then turn them into DIPG. But because it's found in so many of this tumor, more than 70%, we think it's called a founder mutation, which therefore sort of sets the stage for them to become and gain more mutations. So the hope from this research is that we'll be able to figure out how this mutation actually brings about this change in these cells and potentially identify a way that this could function as an Achilles heel. If this project is successful, we will be able to identify potential downstream targets that we could then hit, sort of, as I mentioned, the Achilles heel that we're hoping to find. So this mutation may create new opportunities for us to kill this type of tumor in ways that we would not be able to otherwise find without identifying this mutation and putting it into these cells.